Welcome back. In this video, I will discuss the darkfield microscope, its principle and its applications. In my previous video, I talked about the Brightfield microscope. I have also explained that how contrast is generated in Brightfield microscope. Here, cells absorb some of the direct light, making them appear darker. Because the light that reaches to our eyes, its intensity is decreased, while the background absorbs no light and appear bright. So when we placed our eyes on an eyepiece, cell looks dark and the background looks bright. But Brightfield microscope has some limitations. However, a major limitation of Brightfield microscope is that cytoplasm is nearly transparent and colorless, which absorb very little light and often look similar to the background, making it very difficult to observe detailed structures unless the cells are fixed and stained. These chemicals and heat treatments kill the cells, which makes it challenging to observe certain properties of living cells such as movement, phagocytosis, and cell division. We can overcome this problem by modifying certain characteristics of the Brightfield microscope. How? Let's talk about one major discovery. In 1840, Charles Spencer able to visualize theory of diatom that was never seen before under Brightfield microscope. How he could manage that? He illuminated the object at a certain angle called oblique light, and he captured only the refracted light, which increased the contrast and made the theory clearly visible. Let's take a look at what oblique light is. When you illuminate the object directly, it captures direct light. It looks like this. Now when you illuminate the object from a certain angle called oblique light, shadow is created, and because we are not capturing any direct light, that means the background looks dark than the object. The important fact is that you don't have to stain the cell, but still you can capture a very good image quality. With this setup, the cell boundaries become clearly visible when the light hits the object at this angle. The oblique light effect is the basic behind the discovery of darkfield microscope. Another important discovery was that using this method of illumination, Sweden was able to visualize Treponema pallidum, the causative agent of syphilis. This slender spirochete is almost invisible under Brightfield microscope. Sweden's discovery attracts many biologists and they start using darkfield microscope for medical research. Darkfield microscope became popular after that. Today, darkfield microscope is a crucial optical imaging tool for studying small living objects. The instrument is similar to that of Brightfield microscope, with a visible light source is present here. New introduction is the stopper, which is placed between the light source and the condenser lens. The stopper has a clear central area that allows light to pass through, while the rest of the stopper blocks the light. As a result, the light travels in a hollow cylindrical shape like this. You can try this at your home also. What will happen when a hollow cylindrical light passes through the condenser lens? A hollow cone of light will emerge like this. If we place the specimen in this area, look, no direct light entered inside the objective. Only the light that is reflected or refracted or diffracted by the specimen reaches the objective. Now if we capture a photograph or see directly through the ocular lens, what we can observe? The light is coming from objects only, so definitely the object looks brighter, but no light is coming from the background, so it looks dark. Now the question is, what type of objective lens should I use for darkfield microscope? In darkfield microscopy, the numerical aperture of the condenser lens should be greater than the objective lens. For example, if we are using one objective lens with 0.5 Na, then the Na of the condenser lens should be greater or 0.75 or 1. If you want to know what Na is and how it affects image quality, you can watch this video where I have discussed about numerical aperture in details. Now let's check where darkfield microscope is used. It is generally used to observe those microbes that are very slender, and due to that, it is hard to visualize them under Brightfield microscope. But if we use the darkfield microscope, it is easy to visualize them and clearly, like Treponema, Borrelia, Leptospira in clinical samples. Cellular processes like cell division, cell movement, phagocytosis, and cellular projections like flagella, can often be seen in unstained cells through dark-field microscopy. It is used to observe internal structures in larger eukaryotic microorganisms like stereo of diatoms, as I have already mentioned. Is there any advantages of dark-field microscopy? The answer is yes, because the resolution is somewhat better than that of bright-field microscope, especially at the periphery of the cell when unstained. It improves image contrast without staining, so living cells can be studied. We can also observe the presence of pathogenic bacteria directly from the patient sample, if the bacteria do not grow under laboratory conditions. No rigorous and time-consuming staining process is there, which decreases the sample preparation time. And nowadays, the most of the bright field microscope available in the market can be easily converted to the dark field microscope. But the dark field microscope has some limitation also. When you want to study motility of the microbes, the specimen must be examined as quick as possible. Otherwise, the microbes may die if the sample is not stored appropriately for a longer duration. Only the refracted light is entered into the objective lens, and therefore definitely the intensity of the image will decrease. In order to get a clear picture, the light intensity is increased that may damage the cells. Sometimes debris and artifacts can also scatter light and appear bright that could affect the image quality. The sample should be spread uniformly to get a monolayer. Thick smear may compromise the image quality. Now let's check the similarity between bright field microscope and dark field microscope. In both cases, visible light is used to illuminate the object. Magnification is achieved using the glass lenses, and highest magnification is 100x for objective lens and 20x for the eyepiece. The resolving power is 0.2 micron like all other light microscope. Now let's check the difference between dark field and bright field microscope. The contrast is generated due to absorption of direct light by the specimen, whereas background looks brighter in case of bright field microscope. But on the other hand, only refracted light from the specimen is collected, but from the background, no light is coming. And as a result, the background is darker and the object looks bright. Bright field microscope is routinely used for the laboratory purposes, but on the other hand, dark field microscope is used to observe live and unstained cells only. In order to increase the contrast in bright field microscope, stains are used that are not required in case of dark field microscope. That means sample preparation is easy and direct. That's all for now. I hope you enjoyed this video. If so, please hit the like button and subscribe my channel if you are watching my videos for the first time. And finally, thanks for watching.